Victoria, you've still got a mic on, so
Okay.
we'll let you use that one. And again, just click the green light, give it a second. Online folks, get your questions into the Q&A pod. Let's start here with question or microphone number 2, nice and close for me, please.
Okay, is it on?
It is.
Okay, ICH says months. OGD says 60 days or 90 days for stability. When do we switch over to months after the 180 days? Do we do it at 9 months, 12 months, or does OGD have 5 days less of the year than the rest of us?
You really shouldn't. Is that on?
It's on. Go ahead.
Yeah.
Just a little closer, if you would.
The idea is that if you use months, we run into or if you use days, weeks, we run into a problem of consistency in our evaluation. I know it's just, a day or 7 days or whatever the total comes out to be, but we would like you to say that you use months. We've got a 30-day standard, if you will. It's not really a standard, but it makes it a lot easier to maintain consistency in what people are looking for. We did look at this extensively a couple of years ago, and we just decided that counting the weeks, depending on when the weeks were during the year, we just kept coming up with different extents. And so we've said it's a standard month, which is just 30 days.
Thanks, Frank.
Microphone number 1, please.
I have a question regarding the lots of API that are being used. We are required to use 2 distinct lots of API per strength, 3 batches per strength. And I use 1 lot of API for one of those batches. And for the second or the third batch of the same strength, use 2 distinct lots of API. I'm not pre-mixing, I'm just adding 2 distinct lots of API. Would that still fulfill the requirement of 2 lots of API per strength?
There's been a lot of confusion about what distinct or separate or all of the other words that we've tried to make a clarification. We finally said if you use 1 batch for 1 string, for one batch of API for 1 batch of product, a second batch of API for a second batch of product, you can do whatever you want to for the third batch of product. But we want for at least 2 of the product batches within a strength. We don't want duplicate or mixed API.
Thank you.
An online question, please.
Sure, this message is for Frank. What would be sufficient justification for not including multiple orientations on liquid product stability?
So let me repeat that. I think the question is, when could you not provide information on more than 1 orientation of the product, packaged product? That would be, let me step back just a second. The reason for having this recommendation in the first place is to evaluate the effect of the packaging, the immediate container components on the stability of the product. So if there is no difference anywhere in the container, then it doesn't make any sense to require turning it over if the product is not going to experience a different environment. So that would be one case. And although I hate to say this one, if you could justify that there would be no different environment based on the particular container system, then that might be justification. I think that would probably have to go to the reviewer, though, to make that final determination.
Just to add to that, sorry, so there are some exceptions, and since we don't cover every single case in our guidances, these questions come up. For example, for a blow-fill seal container, do we expect all orientations? What we are looking at from you is just a justification. You can say, hey, the blow-fill seal container that I'm using is just comprises of a single material. So I'm just going to submit, my worst case orientation, and that's okay. For a lyophilized drug product, you can say, hey, the drug product does not contact the closure. So that's another justification. For a large volume parenteral, you can say that the solution is in contact with all the ports. So again, my non-worst case orientation is just one. So we are looking at the product understanding from you, looking again for you to convince us and show us that you understand everything, and there's a reason why you're only providing one orientation. So.
Excellent. Microphone number 2, nice close for me, please.
Yeah. This question is to Dr. Anand. So we are receiving few queries on the discrimination of the particle size for the dissolution method. For some of the products, even trying within the possible limits of the particle size of the API, the dissolution method is behaving the same way. We are not able to exhibit the discrimination. So with that justification, is it fine, and what is your expectation on that query?
The way I understand the question is, you have a method, dissolution method. It is not discriminating with regard to the drug substance particle size, right?
Yeah, that's what the query states.
Okay.
The deficiency states.
So when we evaluate the discriminating ability of the dissolution method, we try to see complete picture. And there are cases when it's not possible to have a discriminating ability with regard to all CMAs, CPPs, right? And this may be one of the cases, right? So the main question is how you are going to mitigate the risk associated with the change in drug substance, right? So if you have appropriate or tight drug substance particle acceptance criteria in place, and they can be justified well, I think that should be okay.
Thank you.
Microphone number 1, please.
My question is directed to the 2 FDA people who talked about genotoxic impurities. Although the M7 does discuss how to classify an impurity as to mutagenic or non-mutagenic with or without carcinogenicity support, and the guidance also only accepts the Ames assay as the critical evaluation of that mutagenicity, what is the agency's opinion of a non-mutagenic impurity whose carcinogenic potential can still be there because there are non-mutagenic carcinogens? How do we evaluate our impurities from that perspective?
I can answer that. So Hongbio had mentioned that Ames Test was kind of the only thing that we look at, but that's actually not true. Whenever an Ames Test is submitted, it's kind of like the first evaluation of an impurity. We get a consult to the PharmTox team, and we actually look at a collection of data. The Ames assay isn't the end-all, be-all evaluation of it. We look at CAR-C data and many other in vivo and vitro assays.
If there is no CAR-C data, having been somebody who submits QSARs to the agency, usually we submit a more robust assessment in which we use all the genotoxicity assays and the carcinogenicity evaluation in order to supply you with a weight of evidence approach that helps you define that. But the agency does not acknowledge any of these assays as being validated or for us to submit it. So it's only on our initiative.
Right, so it's not a requirement of a generic application. Yeah, I mean, any animal studies or in vitro studies related to farm tax issues, genotoxicity are not required in an ANDA currently. It would be upon you to do an initial assessment of your impurity to determine if there is any potential risk of being mutagenic, and then if it's positive or equivocal in silico prediction, then you would follow up with an Ames, and then if you're not even sure about that, the results of your Ames, or if your Ames are positive, then it's on you to decide how to proceed and what test you would conduct to confirm or deny whether it's suspected to be mutagenic.
All right, online question, please.
This question is for Nusie. Is a 2-point intermediate stability 0 through 6 still required in addition to the 3 or 6 months of accelerated for filing?
The question is, do we need 3 months of intermediate stability study in addition to accelerated, assuming that your accelerated study is passed? So the answer is no. Only when you see a significant change in any attribute is when we expect you to provide us with the intermediate data.
Okay, microphone number 2, nice and close for me, please.
Hi, this question is for Dr. Om Anand. I have a question regarding your slide 27 of your presentation, where you talked about IVRT methods for semi-solid dosage forms. So are the IVRT methods used interchangeably with the dissolution method, or when we submit an ANDA, the agency expects us to submit 2 distinct methods, one a dissolution and another an IVRT method?
My understanding is for semi-solids, it's IVRT method, it's no dissolution method.
So once we submit an IVRT method, there is no requirement to submit a separate dissolution method, is that correct?
So if dissolution is not part of your drug product specification, the answer is yes. Okay. So, but what I was saying is, even if that is the case, even if dissolution or drug release in this case, IVRT, is not part of your drug product specification, you may still need it later on for SUPEC kind of changes. And you may end up developing method that time, and that is perfectly acceptable. That's how it is being done. What I was saying is, it is beneficial if you can develop that method in advance, validate it. You don't have to necessarily use it as part of drug product specification. You can develop it. The benefit is you can evaluate your bioequivalence batch or clinical batch at that stage and draw their baseline, which can be used later on for comparison purposes. So that's what I was saying. But if it is not part of drug product specification, there is no requirement like that.
Thank you.
Microphone number 1, please.
Hello. I have a question regarding drug substance stability, like for a micronized API. The DMF holder has stability data which was generated for the non-micronized drug substance. So would they also require to generate data on a micronized API? Or say if you were to use it for an ANDA where it's the particle size is critical?
Here. Sorry, let me just check my notes here. That is an interesting question.
I'll just make a couple of general comments about that. You shouldn't assume that the properties of the response to the environmental conditions for micronized substance are the same as a non-micronized substance, simply because the surface area is, for one thing, considerably greater, sometimes immeasurably greater. And so the idea that using the data generated from a larger particle to cover for the micronized particle is not a good idea. I think you would be asked for both. If you submitted one and if you submitted the larger particle and proposed using the micronized particles, then you would be asked for additional stability data from a typical reviewer, in my mind.
On that, if you don't mind me asking, like, if we had to, like, how many batches would be needed, like the standard 3 batches, or we could just do a single batch?
I'm actually going to intercede, and this is going to be a continued conversation during the lunch break so that they have time to consult their notes. Let's go to an online question if we could, please.
Sure. This question is for Dr. Anand. You mentioned you did not want to see data using expired or aged products, but what if that is the only type available to you? For example, the manufacturing ceased at the previous manufacturer over a year ago.
Well, that happens sometimes, and you can provide your justification. But for comparison purpose, what I would do is go back a little further and look into my files. And instead of using expired pre-change batch data, I would try to see how the bioequivalence batches are performing. And if you can submit that data with addition to the data that you're generating right now, I think that will be very helpful. And if you have questions, then we can begin the conversation. And we can determine based on the totality of information that is available at that time.
Microphone number 2, please. Nice close.
My question is for Dr. Holcombe. You showed in your presentation some figures and percentages of the RTRs due to having less than 180 days of stability data. And you showed the figures from fiscal year 2015 until now, and it's kind of gone up. So I was just wondering, from your opinion, what do you think that's indicative of?
I'm not sure I understand the question. Are you asking what's the significant where the percentages rise over time? I don't think I can answer that. There are a lot of factors that go into what we use as a standard, and sometimes they're very simple factors, such as we may change words in a guidance and industry misinterprets the change that we've made. In other cases, there may be an influx of new companies that just have not been familiar with the practices. So I think the one thing that you might call significant is that if the quantity or percentages continue to increase, that is an area that we at the agency need to look at to see if we need to readdress the documentation that we've already put out.
Thank you.
Microphone number 1.
I wanted some clarification regarding the stability requirement for a new ANDA. If I have 24 months of passing long-term stability data, am I still required to submit the 6 months accelerated stability data with the new ANDA, or will the 24 months suffice to support the stability requirement?
So you're asking if real-time data would be sufficient, right? So at the time of filing to get into the door, it would be sufficient. So this would buy you additional time by the time you can submit, a gratuitous amendment, or if you receive some deficiencies. So you can get into the door with your long-term real-time data, and then you have a long time to generate the 6 months of accelerated data to support the review of the application, the technical review, in case a reviewer.
If I have the accelerated and the long-term available,
Yeah.
I would have to then submit both to not delay. My question is basically, how would that affect an RTR if I'm only submitting the 24-month long-term? I already have the 6 months available. So I wanted to know if it is a requirement to have both, or if the real-time data, it's just necessary.
So again, it depends on your actual drug product. So to get into the door, the long-term would be sufficient if it's a stable dosage form and the examples that I provided. So if it's in a refrigerated or a liquid, then you will need accelerated at the time of filing. Otherwise, it'll be RTR.
Thank you.
All right, we've got time for about 2 more questions. Let's take an online question, please.
For a non-traditional dissolution method, a custom method such as a dialysis membrane, the inherent variability is higher, approximately 10% due to the variability in membrane for a complex drug product. Is this acceptable by the FDA, for the FDA?
Well, at this stage, I would not say that it is acceptable, but we can definitely discuss it. If you provide all the information, we are open to discussion and take it from there.
Microphone number 2, please.
This question is for Dr. Anand again. It has to do with dissolution studies to determine dose dumping. From what we understand, and in the absence of a specific guidance related to this topic, that when the RLD were asked to do dose dumping studies, the generic will have to generate the similar data. In the absence of any such requirement for the RLD, but when a follow-on RLD, basically it's the same product with a similar, basically with the same ingredients, but a follow-on product that was requested dose dumping studies. So if the original product-specific guidance was not updated to require specific dose dumping studies, is it safe to assume that that is not required, or should we ask the question?
Is it to alcohol dose dumping?
Yes.
Okay. So we in Division of Biopharmaceutics do not review that. The question should be directed more to the Division of Bioplans, Dr. Kiran, because they review that part of the dose dumping studies. So I'm not in position to comment on that.
Okay, so we should be directing that question to CMC or the quality group?
No, that question should go to Division of Bioplans OGD.
Thank you.
Okay, and I apologize to the folks waiting at the microphones and the dozens and scores of questions that we had come in online. We are running a little bit long. I want to make sure that we can get to lunch and have time for all our topics this afternoon. So please help me thank our panel for coming and sharing their wisdom with us. We will extend, since it's